Today we're going to have a look at cell division. Both of these come up in biology. Mitosis comes up in paper 1. Meiosis comes up in biology paper 2. So we're going to have a quick whiz through. It's just for revision now. For mitosis, this is the cell cloning that takes place in any body cell. So we put cloning up the top here. Now essentially what we're doing is we're starting here with the original DNA that cell has. It might be a cell in your kidney, it might be a cell in your liver, it might be a cell in your skin or in a bone, and this one cell has to copy itself completely and create identical cells, 2 copies of itself. Now the process of that, mitosis, I always think about the word toe and the idea that your body cells, including your toe as a body part, any type of cell division that comes up in the exam is most likely to be about mitosis. Okay, so in paper 1 you need to understand that one single cell, if it's a human cell, will have 46 chromosomes in, and that means there are 2 different copies. One that you've inherited from mum, the maternal chromosome, one that you've inherited from dad, the paternal chromosome, and because there's 2 copies, that means it's diploid, so di meaning 2 and ploid meaning chromosome, so 2 copies of each chromosome. Now ultimately what you'll see down here are 2 new cells, what we call daughter cells, and each of them have the original DNA in there, but they have a copy of each chromosome as well. So these 2 here, those chromosomes have actually been separated into 2, one in each cell. The smaller chromosome here, we can't draw all 23 pairs, so just sticking with 2 pairs for now. One copy has gone to one cell and the other side has gone to the other cell. Now the idea here, the very first thing that happens is your DNA is replicated. So before any cell division happens, the contents of the nucleus have to be copied. You've got your DNA copied down here, and you'll notice our original DNA is paired up with some new DNA. So we've got double the amount of DNA in here now, 92 chromosomes, just for a short period of time, before they get separated to opposite poles of the cell. I like to think about Spider-Man being in the corner over here. It's actually called a centrosome. And Spider-Man's in the corner, and Spider-Man spins his web across the cell, and these things are called spindles. So a bit like spider silk, which are made from protein. Spindles are made from protein. Spider-Man this side, Spider-Man that side are going to pull the correct copies of the chromosomes across to the opposite poles. That way, the right chromosomes end up at both sides of the cell, over here, and then they therefore can separate and form 2 nuclei in 2 different cells, each with an identical genetic makeup to that original cell. Okay, so our period of time where we've got double the amount of DNA, we've got our spindles pulling the chromosomes across, Spider-Man pulling across the chromosomes to opposite poles, and once that's done we've got 2 daughter cells which are diploid, so both of them have the full complement, 2 copies of every chromosome. Key points about mitosis that you need to know: it's all about cell cloning, so genetically identical daughter cells, and that it occurs in all body cells. Bacteria are going to be doing the same thing, plant cells are going to be doing the same thing, mitosis, as they grow new tissue. That's for them is their body cells. So there's mitosis, that's the paper 1. Then moving on to the paper 2 part, this is meiosis. Now if you're wondering where the cells actually came from to start with here, that has to come from the process of fertilisation, and to do that you've got to have 2 cells that have half the genetic material, so just 1 copy of each chromosome. That means 23. Now if we skip right down to the bottom here, these are the 4 cells that are going to be produced from mitosis, and these are gametes, so they are sex cells. They're either sperm cells or they're egg cells, or they might be in a plant pollen cells for the male gamete in a plant. So 4 cells. Notice instead of the 2 copies of each chromosome, each of them have 1 copy of each, so half the genetic content, hence where we get the word haploid from, meaning half the number of ploids, or half the number of chromosomes. So we start with the same cell, we've got 46 chromosomes. The key for this is slightly different. I'm using black here to represent paternal chromosomes. That's the chromosomes that this person has inherited from their dad, and the orange colour is the chromosomes that this person has inherited from their mum. So each one of the 23 pairs that make up 46 chromosomes, you have one copy from mum, one copy from dad. And the first round that happens here is that one cell undergoes a full round of mitosis. So you get a complete copying, a cloning of this one cell to become two. And that is the first stage of meiosis. The difference comes now where these two cells have to divide in half. So the DNA has to be split to form 4 cells, each with half the number of chromosomes in. In order to do that, a bit of magic happens, and this is where this process is really special. What I've done with the colours here is to try and show you that the paternal chromosomes and the maternal chromosomes actually get snipped by enzymes, and they get transferred across to each other in completely different ways every time a cell is copied. So you'll notice here we've taken this and we've snipped this chromosome into 2, and the top halves we've just swapped over. So this cell here has got swapped DNA across. Likewise over here, but we've snipped it in 2 different places. We've just transferred the tips at the top and the tips at the bottom and swapped those across. Now you've got chromosomes that have got some of dad's DNA in, the paternal DNA, and some of mum's DNA, the maternal chromosomes, in here as well. Likewise, the same thing's happened over with this cell, but it's just been snipped at different points. Now you see these 2 cells are already very different in the combination of genes that they have on each chromosome. If you then undergo the next round of cell division, where each cell ends up having half the genes in, then one of these big chromosomes and one of these little chromosomes is going to end up in one cell. So you've only got one copy. Perfect, because this sperm cell or egg cell is going to need to combine with a sperm cell or an egg cell to create a fertilised egg. That will need, of course, 46 chromosomes in. So we only want sperm and egg with 23 chromosomes in. So we can see, if you map each of those and just have a look at those there, you'll see that all 8 strands of DNA here, all 8 chromosomes, are present down the bottom. But every single one of these has got its own unique combination of that original DNA that this person has inherited from their dad and from their mum. Every sperm cell and every egg cell is genetically unique to any other egg cell or any other sperm cell that that person will ever make. So the result of meiosis is 4 haploid gametes, each with 23 chromosomes, as opposed to mitosis, which has only 2 cells made, each with 46. Now, if you're wondering about what sort of things might come up in the exam for mitosis and meiosis, I've put a few questions up here. You might want to press pause, draw a little table out and have a go yourself, and then press play in just a minute so I can go through the answers with you. All you've got to do is tick a box. So if we look at the question number 1, which of these occurs in body cells? Now that's mitosis. I always think of mito being a body part, and therefore any body part, whether it's skin or bone cells, kidney cells, liver cells, they're going to undergo mitosis to generate new cells. Producing gametes, now these are sex cells, is meiosis. And the little trick I remember with that is we've got an E for egg in meiosis, and we've got an S for sperm in there. So egg and sperm means meiosis. So that's only going to take place in the ovaries and the testes in animals. Involves DNA replication. Well, you can't have any cell division without DNA replication, so that's going to be both. Occurs in the testes or the ovaries, that has to be meiosis. Results in diploid cells, di meaning 2, ploid meaning chromosomes, so 2 copies of chromosomes. The cells produced with 2 copies are going to be normal cells for humans with 46 chromosomes in. With haploid cells, that's short for half if you like, and ploid, so that's half the number of chromosomes. That would be 23 in humans, and that, of course, is the gametes. Then we have genetically identical cells. That's essentially a clone. So genetically identical cells is mitosis. And unique cells will be meiosis. That's sperm and egg cells with their own unique genetic combination. Well, the best thing about meiosis is the idea that you get variation, and I should really have the word biodiversity on here, because with all these different combinations of genes that each individual inherits being combined together to form new combinations, every time a sperm cell or an egg cell is made, you get the opportunity for variation in characteristics. Now, the other thing you might be aware of, just pop a sequence together of pictures. I'm hoping that's come through on the video okay. From start to finish with meiosis, you might be asked to identify which cells might be undergoing meiosis or not. Now, essentially, all you're doing is looking for cells with a nucleus. If there's a defined nucleus, you can see the genetic content inside the nucleus, particularly if it's all wrapped and packaged up nicely. Then it's unlikely that that cell is undergoing mitosis or meiosis. It's really when you start seeing the chromosomes start to shorten and start to thicken, and then start to line up along the middle in metaphase here, along the equator of the cell, and then they start to be pulled apart by the spindles you can see here, forming two, or about to form two separate nuclei. These are the characteristic stages of cell division. So that's really what you're looking for, is the DNA, the chromosomes, being packaged up, lined up along the equator, and then pulled across to the opposite poles of the cell.